Welcome to the PubMed Daily Review for April 23rd, 2026. Today we scanned 6,269 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, The Transformative Impact of AI-enabled AlphaFold 3, Evolution, Current Status, and Future Prospects in Structural Biology by Chiranjeev Chakraborty and Sang-Soo Lee from Department of Biotechnology, School of Life Science and Biotechnology, Adamas University, Kolkata, India. Let's break down its key contributions. For decades, understanding how proteins fold into their complex 3D shapes has been a major challenge in biology. While experimental techniques exist, they are often time-consuming and can't solve every structure. This is where artificial intelligence, particularly Google DeepMind's AlphaFold, has made a transformative impact, dramatically accelerating our ability to predict these vital molecular structures. AlphaFold's journey began with AlphaFold 1, which used deep neural networks to predict protein structures. AlphaFold 2 significantly improved accuracy using its eviformer architecture, achieving near-experimental precision. The latest version, AlphaFold 3, introduces the Pairformer module, expanding predictions to include protein ligand, protein nucleic acid, and protein-protein complexes, marking a new era in biomolecular modeling. The impact of AlphaFold has been profound. Structural coverage of the human proteome surged from 48% to 76%, while the dark proteome, regions with unknown structures, shrank from 26% to just 10%. The AlphaFold Protein Structure Database now hosts over 214 million structures, democratizing access. Notably, AlphaFold 3 demonstrated a 50% improvement in predicting protein ligand binding positions compared to traditional methods. AlphaFold advancements are revolutionizing drug discovery and our fundamental understanding of biology. While challenges like predicting protein dynamics persist, the trajectory is clear. AI-powered structural biology is poised to drive significant progress in biotechnology and medicine, opening new avenues for research and therapeutic development. Up next, our second paper out of 5. Single-cell multiomics reveals regulatory mechanisms of CAR T-cell persistence and dysfunction in multiple myeloma by Lorea Jordanariza and Felipe Prosper from Hemato-Oncology Program, CIMA Universidad de Navarra, EDSNA, Pamplona, Spain. Let's break down its key contributions. Chimeric antigen receptor, CAR, T-cell therapies have revolutionized the treatment of blood cancers like multiple myeloma. While these therapies can induce deep remissions, many patients eventually relapse. A key challenge is understanding why CAR T-cells sometimes lose their effectiveness over time. This study aims to uncover the underlying molecular mechanisms that govern CAR T-cell persistence and dysfunction in the context of multiple myeloma. To tackle this, researchers employed advanced single-cell multiomics techniques, analyzing both RNA and T-cell receptor sequences from CAR T-cells. They studied cells collected from patients with multiple myeloma at different time points after infusion, comparing cells from bone marrow and peripheral blood. A key part of their approach was using gene regulatory network analysis to understand the complex interactions controlling CAR T-cell behavior. This allowed them to pinpoint specific cellular states and molecular pathways involved in CAR T-cell persistence and dysfunction. The study revealed significant differences between CAR T-cells found in the bone marrow compared to those in peripheral blood. Bone marrow CAR T-cells exhibited a more activated and exhaustive profile. They observed that a dysregulation in the normal transition from effector to memory cell states led to an increase in terminally differentiated CAR T-cells, which correlated with poorer persistence in patients. Furthermore, in one patient with a partial response, a specific clone of CAR T-cells producing IL-10 was found to be expanded, and this IL-10 production was amplified when the T-cells were stimulated through both the CAR and their endogenous T-cell receptor. These findings highlight critical mechanisms that limit CAR T-cell effectiveness in multiple myeloma, particularly the exhaustion and impaired memory development within the bone marrow. The identification of IL-10 producing clones suggests a specific pathway that might be exploited for therapeutic benefit. Ultimately, this research provides valuable insights for developing strategies to improve CAR T-cell persistence and overcome resistance, potentially leading to better long-term outcomes for patients with multiple myeloma. Up next, our third paper out of 5. Targeting Apolipoprotein E4 rescues photoreceptor degeneration by inhibiting the disease-associated microglia activation in experimental retinal detachment by Xiaoming Li and Kai Dong from Ophthalmology, the first affiliated hospital of USDC, Division of Life Sciences and Medicine, University of Science and Technology of China, Hefei, Anhui 230001 People's Republic of China. Let's break down its key contributions. Retinal detachment is a serious condition that can lead to irreversible vision loss due to photoreceptor cell death. In the retina, specialized immune cells called microglia can become activated in response to injury, forming a disease-associated microglia or DAM phenotype. While this activation is linked to the disease process, the specific role of Apolipoprotein E4 in this process and its potential as a therapeutic target in retinal detachment remained unclear. To investigate this, researchers used a mouse model of retinal detachment. They employed genetic tools, including APO knockout mice and mice engineered to express specific human APO isoforms like APO4. Advanced techniques like single-cell RNA sequencing and Western blotting were used to analyze microglial activation and photoreceptor survival. The study aimed to pinpoint how APO influences the DAM phenotype and whether targeting APO4 could protect photoreceptors. The results were quite striking. In mice expressing the APO4 isoform, there was a significant improvement in photoreceptor survival following retinal detachment compared to wild-type mice. This protective effect was directly linked to a significant suppression of disease-associated microglia activation. Interestingly, while APO deficiency also showed some protective effects, the APO2 and APO3 isoforms did not confer the same benefits as APO4. This study highlights a promising therapeutic avenue, targeting Apolipoprotein E4. The findings suggest that APO4's ability to inhibit detrimental microglial activation could be harnessed to protect photoreceptors in conditions like retinal detachment. Modulating the APO-galectin-3 pathway presents a potential new strategy for developing treatments. While these results are encouraging, further research is essential to confirm these benefits and explore their translation into effective clinical therapies for patients. Up next, our fourth paper out of 5. Spatial transcriptomics unveils immune cellular ecosystems associated with patient survival in diffuse large B-cell lymphoma by Alba Diaz-Herrero and Pierre Tanner from A Institut de Recherche St. Louis, University Paris site, INSERM U 1342, Paris, France. Let's break down its key contributions. Diffuse large B-cell lymphoma, or DLBCL, is the most common type of non-Hodgkin's lymphoma. While treatments like R-CHOP have improved outcomes, a significant portion of patients still relapse or don't respond. A key reason for this is the complex and varied immune microenvironment within the tumor. We need to understand how the cells are arranged in space to better predict who will do well and who might need different treatments. To tackle this, the researchers used spatial transcriptomics on DLBCL tumor samples. This technology maps gene expression directly within the tissue, allowing them to see where different cell types are located and how they interact. They analyzed 10 DLBCL samples and some control tissues. By applying advanced computational methods, they identified 6 distinct spatial patterns or cellular ecosystems within the tumors. The study revealed 6 distinct cellular ecosystems within the DLBCL tumors, each with a unique mix of immune cells and spatial arrangement. Importantly, they developed gene signatures based on these ecosystems. When tested on larger patient cohorts, these signatures were able to robustly stratify patients based on their survival outcomes. This highlights that the spatial organization of the tumor microenvironment is a critical factor in determining prognosis, even more so than just the overall number of immune cells present. This research demonstrates the critical importance of spatial context in the DLBCL tumor microenvironment. The identified cellular ecosystems and their associated signatures offer a novel way to predict patient outcomes. This spatially informed approach could lead to the development of new prognostic tools and potentially guide the design of future therapies that specifically target the tumor's spatial architecture or the interactions within these immune niches. Up next, our 5th paper out of 5, Labeling and Imaging of Neurons with a Genetically Encoded Autonomous Bioluminescence System by Teresa Brinker and Carola Greger from Optical Nanoscopy, Institute for Nanophotonic Göttingen e.V. 37077 Göttingen, Germany. Let's break down its key contributions. Visualizing neurons is crucial for understanding the nervous system. Traditional fluorescence methods have limitations for long-term studies due to light-induced damage. Bioluminescence, which generates light internally, offers a solution. However, most bioluminescent systems need a chemical substrate added from the outside, which can be inconvenient and limit imaging duration. This study aimed to develop a fully autonomous bioluminescence system for neurons. To achieve autonomous bioluminescence in neurons, the researchers engineered specialized adeno-associated viruses, or AAVs. These AAVs were designed to deliver the 6 genes required for the bacterial bioluminescence system directly into neurons. They optimized the viral packaging and promoter selection to ensure bright, neuron-specific light emission. This approach allowed for long-term, real-time monitoring of neuronal activity in response to stress without needing any external chemicals. The developed system achieved a significant bioluminescence signal, about 35% as bright as the standard Firefly luciferase system. Importantly, the transduction efficiency reached 44%, meaning a substantial portion of neurons were successfully labeled. The system demonstrated excellent stability, with signals remaining consistent for up to 20 hours. Crucially, the bioluminescence signal effectively decreased in response to neurotoxic agents like hydrogen peroxide and glutamate, indicating its utility in monitoring cellular health and stress. This study presents a significant advancement in neuronal imaging technology. The autonomous bioluminescence system offers a powerful, non-invasive method for long-term observation of neurons without the need for external substrates. Its ability to detect cellular stress in real time makes it a promising tool for investigating the mechanisms of neurodegenerative diseases and for screening potential therapeutic interventions. This technology could accelerate our understanding of brain function and disease progression. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.